Hi guys, let's do section 4, the nucleus and ribosomes. The nucleus is the command center of the cell. It's usually found near the center. It is separated from the cytoplasm by something called a nuclear envelope, which consists of a double layer of membranes. It has pores called nuclear pores that allow the exchange of material from the inside the nucleus and the cytoplasm. The nucleus contains chromatin, and a semifluid called nucleoplasm. The chromatin contains nucleic acids and proteins, which condenses to form chromosomes. Chromosomes are formed during cell division and are the carriers of genetic information. So chromatin is just long strands of DNA. The dark nucleolus is composed of something called rRNA or ribosomal RNA, and it produces the subunits that make up a ribosome. Here is the anatomy of the nucleus. So here is a drawing. Okay, here we see the nucleolus, which is a dark region where RNA is made that makes up ribosomes. Down here, this stringy stuff, that's chromatin. The lighter purple makes up that nucleoplasm. Okay, this outer layer, the smooth bit here with the holes in it, that is the nuclear envelope. Here we have a zoomed in look at it. It's tiny little holes that allow things like RNA to go in and out. If we were to zoom in even more, we would see the phospholipid bilayer within that, or that makes up that membrane. Okay, so we've got our glycerol heads here and our fatty acid tails. Remember the kinks in it. The bends are from double bonds that make it more stable and able to withstand temperature changes. Here we have an actual image of a nucleus. You can see the tiny pores in the surface that allow RNA to go in and out. Ribosomes. They are composed of rRNA or ribosomal RNA. They consist of a large subunit and a small subunit, which are made in the nucleolus. Ribosomes may be located on the endoplasmic reticulum, which is an organelle that we're going to talk about later on, or they can be found free-floating in the cytoplasm, either singly or in groups called polyribosomes. These RNAs are the site of protein synthesis. This is where protein gets made. The process of that is called transcription and translation. Transcription takes place inside the nucleus. It's when a copy of DNA is made with just the important bits that then travels through the pores in the nuclear envelope, through the cytoplasm to a ribosome, where it is translated or read by a ribosome, and the ribosome then puts together a strand of amino acids, which, with the help of chaperone proteins, is folded up into a functioning protein. So I basically summed up these 3 bullet points, but I'll go ahead and read them for you guys, just in case you're wanting that. Information for the gene is copied onto an mRNA, which is then exported into the cytoplasm. Ribosomes then receive that mRNA with a coded message from the DNA with the correct sequence of amino acids to make a protein. Proteins synthesized by cytoplasmic ribosomes stay in the cytoplasm. Those attached to ribosomes end up in the ER. The central dogma of molecular biology is the DNA goes to the mRNA to the protein. So we start with the DNA in the nucleus. We make a copy of RNA. That mRNA then goes to the ribosome and makes a protein. Here we have a diagram showing how a ribosome reads the mRNA strand. So here's the mRNA strand that has been read. It has gone through the ribosome, and the ribosome takes the information on that mRNA and puts together a strand of amino acids. So right here we have a primary structure of a protein. it will begin to fold up into the secondary structure, whether that's an alpha helix or a beta sheet. I don't know from this diagram, it's impossible to tell. With the help of chaperone proteins, it will be folded until it reaches a fully functioning structure. And that's what I got for you guys on this section. If you have any questions, feel free to shoot me an email.